Hello everyone, I will introduce an article published on iMeta. Biochar stimulates tomato roots to recruit a bacterial assemblage contributing to disease resistance against Fusarium wilt. Dr. Shuai from Northeast Agricultural University is the first author. Professor Xingang Zhou from Northeast Agricultural University and Professor Zhong Wei from Nanjing Agricultural University are the corresponding authors. Introduction. Plant diseases cause serious economic losses in agriculture. Biochar amendment is acknowledged to control plant diseases. Plant rhizosphere microbiome is a key determinant of plant health and productivity. Biochar amendment can alter the composition and diversity of microbial communities and stimulates the abundance of PGPRs in the rhizosphere. However, the mechanism behind this stimulating effect remains unelucidated. Root exudates govern numerous rhizosphere processes, including the recruitment of PGPR and shaping of the rhizosphere microbiome. Therefore, it is possible that biochar amendment can increase the abundance of PGPR in plant rhizosphere through the host plant. Results. In the pod experiment, the biochar treatment promoted tomato growth, decreased tomato Fusarium wilt disease index and FOL abundance in tomato rhizosphere as compared with the non-amended control. The transplanting rhizosphere microbiome experiment found that, as compared with the treatment inoculated with non-sterile rhizosphere soils of tomato seedlings grown in soils non-amended with biochar, the treatment inoculated with non-sterile rhizosphere soils of tomato seedlings grown in soils amended with biochar decreased Fusarium wilt disease index and rhizosphere FOL abundance. Illumina-based 16 ribosomal RNA gene amplicon sequencing found that biochar altered tomato rhizosphere bacterial diversity and community composition. Particularly, biochar amendment stimulated the OTU 1633, belonging to Pseudomonas species. Quantitative PCR analysis found that biochar amendment increased the abundance of Pseudomonas species in tomato rhizosphere. Culturable Pseudomonas species in tomato rhizosphere were isolated. The isolate TP27, which displayed 100% sequence similarity with OTU 1633, showed in vitro antagonistic activity against FOL on PDA agar and decreased Fusarium wilt disease index in a pod experiment. A split root system of tomato was used to test whether biochar amendment stimulates PGPR in tomato rhizosphere via the host plant. Results found that, as compared with the non-amended control, biochar amendment significantly increased the abundance of TP27 in tomato rhizosphere. Lastly, in vitro tests showed that root exudates of tomato grown in soil inoculated with TP2 from the biochar treatment promoted the chemotaxis response and biofilm formation of TP27 as compared with that from the non-amended control. Summary. Biochar enhances the disease-suppressiveness of tomato rhizosphere microbiome. Biochar stimulates tomato to actively recruit plant-beneficial bacterial taxa. This study showed that Biochar-elicited recruitment of PGPR contributed to the enhanced disease suppressiveness of tomato rhizosphere microbiome against Fusarium wilt. Results highlighted the importance of the host plant in this enhanced recruitment of PGPR. Thank you for your attention.